Hello and welcome to this webinar titled Development and Manufacturing of Recombinant Nanoparticle Vaccines, hosted by Biopharma Asia magazine and presented by Atin Tomar, President and CEO at CPL Biologicals BVT Limited, and Amélie Boulais, Vaccine Platform Marketing Manager at Sartorius Stedim Biotech. My name is Orsolya Balog and I will be the moderator. Now, please allow me to introduce our first presenter, Atin Tomar, President and CEO at CPL Biologicals BVT Limited. Atin Tomar, President and CEO of CPL Biologicals, brings with him comprehensive experience in the biotechnology industry globally. Atin's experience and expertise spans from process development, technology transfer and manufacturing operations to business development, licensing and commercialization. Prior to CPL Biologicals, Atin was associated with the biotechnology industry in various roles. He was involved in the development of biologics, biotherapeutics from the early stages of development to commercial manufacturing at Human Genome Sciences, Amgen, and Syngene International. At Dr. Reddy's Laboratories Limited, MSD Wellcome Trust, Hillman Laboratories, and International Vaccine Institute, Atin served in business development and licensing, program management, and alliance management roles. Welcome, Atin.
Thanks, Ursula, for the introduction. Hello, everyone. So I am going to talk about the development and manufacturing of recombinant nanoparticle vaccine platform that CPL Biologicals works on. And before I get started, just a quick introduction to CPL Biologicals. It's a joint venture company established in 2009 between Stedim Pharmaceuticals Limited from India and Novavax from the US. Novavax is a US-based late-stage clinical development company and has developed multiple novel technologies. And as part of the joint venture, CPL Biologicals has access to technologies developed at Novavax as well. So coming to the introduction of the nanoparticle vaccines, we believe that they represent a new paradigm. And I would like to make clear distinction between a typical or conventional definition of a nanoparticle vaccine or product where an vaccine antigen or adjuvant is either adsorbed or absorbed on a synthetic nanoparticle. That's not the case with our platform technology. Our technology is purely protein-based technology where the protein antigens get assembled, self-assembled into a nanoparticle format. So just for clarity, the technology is very different from the conventional definition of nanoparticle products. So what is it that we really do? So the key to develop nanoparticle-based vaccine is first to identify an appropriate antigen, which could be a surface protein, it could be a protein embedded in the cell wall. And once we have identified an appropriate antigen with immunogenic epitopes, we can develop, utilizing our technology, a vaccine candidate. Now, this technology allows us, therefore, to specifically target vaccines against highly immunogenic epitopes and antigens, unlike the conventional vaccines where a whole pathogen, either live attenuated, killed, inactivated, or different components or complex components of the surface of a pathogen are used as a vaccine candidate. So with that, with this technology, all we really need is the genetic sequence of the antigen of interest. Once we have identified the genetic sequence of the antigen of interest, we sequence sequence the gene and express it in SF9 insect cell and baculovirus expression system. Another beauty of this technology is that since we are only working on protein antigens or targets, we can make vaccine candidates even against conserved and hidden epitopes, which can help us make very specific as well as conserved vaccine products. That means that since most of the conventional vaccines work against the antigens on the surface of the pathogen, which evolve with the evolution of the pathogen, while if we can develop vaccine candidates against the conserved epitopes, with such vaccines should be able to provide protection against evolving serotypes of viruses. This technology has been utilized to develop and license 2 vaccines in India. We have licensed H1N1 or swine flu vaccine as well as a seasonal influenza vaccine. And these were the world's first virus-like particle-based influenza vaccines to be licensed anywhere. We've utilized this technology and developed multiple products which have gone into human clinical trials across the globe, and there is safety data of more than 20,000 subjects globally under a clinical trial. And again, since we've recently licensed and commercialized the seasonal influenza vaccine in India, these vaccines have been administered to a large number of population now. Another important aspect of this technology is that it can be applied broadly across various infectious diseases. Since all we are interested in is really a protein antigen, that antigen could be from any pathogen. It could be a viral pathogen, a bacterial pathogen, even parasitic pathogen like malaria. And some of the products that we have worked across infectious diseases are listed here. I mean, we've worked on influenza, RSV, rabies, Zika, Ebola, MERS, among others. We've developed a product for a vaccine for C. diff, as well as done some work on a malaria vaccine. So again, extremely flexible technology to target multiple infectious diseases. Another important aspect of this technology platform is that since we only need the gene sequence of a potential antigen, we can get started with developing a vaccine candidate as soon as we have the genetic sequence for that antigen. It takes about a week to get the clone ready. And given the platform nature of this technology, we can really quickly move into CMC process and manufacturing and potentially produce human or clinical trial material, GMP grade material suitable for human use in a very, very short period of time compared to the conventional vaccines where it could take almost 8, 10, even 12 months to develop a vaccine for a new pathogen. So in terms of the types of nanoparticles we are developing, there are 2 types of nanoparticles that we are working on. As I mentioned, our seasonal influenza and H1N1 swine flu vaccine licensed in India is a virus-like particle-based vaccine. What that means is that we have a viral core, a capsid, and then we express the antigens of interest on the surface of that core. So if you see in the picture, there is a core structure, and then multiple antigens are expressed on the surface. Now, conventionally, the influenza vaccines primarily work on HA protein. There are 3 key antigens in an influenza virus, HA, NA, and M1. However, NA and M1 protein actually get destroyed during the process of inactivation and so on in a conventional influenza vaccine. However, in our case, since it's a completely recombinant technology and there is no virus inactivation required, we are able to successfully express all three antigens on the surface of the virus-like particle. So we have vaccine which actually works against HA, NA, and N1, where N1 is a matrix protein and is believed to be conserved across serotypes. Also, there is publications that NA protein actually provides as much immunogenicity against influenza virus as does HA protein. So basically, our vaccine not only works against HA, like most of the conventional vaccines, or all of the conventional vaccines in fact, we also have immunogenicity derived from NA and M1 proteins. And then, so this virus-like particle format works extremely well in case of a vaccine candidate where we have multiple antigens. So we can express multiple antigens on the surface of virus-like particles. However, in case of a single antigen vaccine candidate, it's important for us to assemble that single antigen, one, to express and stabilize, and then assemble that antigen into a critical sized particle so that our immune system is able to actually recognize and generate immunogenicity. So the technology that we have, what we are able to do is once we've identified the protein antigen through our proprietary technology, we are able to express the antigens, stabilize them in a multimeric format. That means a single antigen vaccine will have the single protein assembled into a multimer. The picture that you see is for an RSV vaccine candidate, where it's a 5-mer. Our rabies vaccine is a 3-mer. The idea being that by assembling into a multimeric format, one, the protein stabilizes itself. Also, a multimeric format gives a critical size to the vaccine candidate so that the immune system can actually recognize and generate immunogenicity. So it's extremely important for us to be able to efficiently express and assemble the protein antigens in an appropriate format to make an effective vaccine candidate. And both of these types of nanoparticles are in the range of, virus-like particles are in the range of 150 nanometers, and the protein cells could be anywhere from 80 to 120 or so nanometers. So they're both nanoparticles in terms of the size definition. Coming to the next slide, the beauty of this process or the technology is that this enables us to actually have a platform technology manufacturing process. So what this means is that especially the upstream process for all of our vaccines is exactly the same. So it's the same equipment, same number of steps, same process, same scale for us to manufacture any of our vaccine candidates, whether they are virus-like particles or protein cells. They all have exactly the same process for upstream. So there is an expansion of SF9 cells, and then the baculovirus cells are expanded into a separate system. SF9 cells are infected with baculovirus, and then the protein or the vaccine candidate of interest is expressed. Now there is a difference depending on if we are trying to make a virus-like particle or a protein cell. The virus-like particle actually bursts out of the cell surface and is purified from the supernatant. However, the protein cells are part of the cell paste and is extracted from the cell paste. So up to the upstream production and harvest, it's the same. And then depending on the nature of this product, we either take up the supernatant or work with the cell paste to purify the product. But for upstream, it's identical process for all of our vaccine candidates. The next slide depicts a standard upstream manufacturing process Again, as I said earlier, it's identical for all of our products, whether it's virus-like particle-based vaccine or protein micelle. So again, we start with a cell bank. We expand the SF9 cells, starting from the vial to shape flask, moving on to a wave bioreactor, and then inoculating a larger scale production bioreactor. Very similar process for expansion of baculovirus, which is again expanded into an SF9 cell bank, and then once a certain concentration of virus or titer of virus is reached, that virus stock is used to inoculate the production bioreactor. All of this process is completely single-use disposable, as you can see. So we are able to quickly turn around the manufacturing facility, not only to move on to the next batch, but also to potentially switch between different vaccine products in the same manufacturing facility. Now, as I mentioned earlier, that up to the harvest, the process is exactly the same for manufacturing. This slide actually shows the downstream purification process for rabies G protein-based nanoparticle vaccine. And actually, there are a couple of, there's one step missing here. After the harvest, there is a cell lysis step. It's a detergent-based cell lysis, which is then passed through a filter and then loaded on to cation exchange chromatography. So chromatography or downstream process needs to be adapted and optimized based on the nature of the specific protein or antigen. As we all know, downstream will depend in terms of the types of resins and chromatography steps that are used will depend on the nature of the protein. However, what we do, let me get to the next slide first. And the next slide is really the purification process for influenza VLP. So basically, from the production bioreactor, we use centrifugation. And centrifugation is actually used for both the nanoparticle, that is the virus-like particle, as well as the protein micelle-based products. And as I said earlier, if it's a virus-like particle, then we just take the supernatant and take it for downstream processing. If it's protein micelle, then the cell paste is of interest to us. So we've actually also implemented a single-use certification system called Kstep, and I think you will hear more about Kstep in the presentation by Amélie later on. And that's a single-use certification process wherein, depending on the product of interest, we can either take the supernatant or the cell paste and take it for further purification. So in case of virus-like particles, since we are working with the supernatant, there is no need for cell lysis or working with the cell paste, so it's run through the filter and then taken up for further purification. And in case of virus-like particle, we have a viral inactivation step because, though baculovirus is a grass virus, it does not infect a mammal, a human, or in fact any mammal. But still, there is a virus inactivation step that we have incorporated into a VLP-based process, which is not required in case of protein micelle because the cell paste is lysed and all the virus is destroyed. This is just for the safety factor, but as we all know, for a baculovirus-based system, there is really no risk of infection to humans or any mammals. So to really summarize the technology and the manufacturing platform, this is an extremely powerful technology with significant advantage where carefully designed constructs really enable us to develop a vaccine candidate against key components of the pathogen. Really highly immunogenic epitopes in a particular pathogen, and that in turn results in enhanced functional immunity and better protection. The SF9 baculovirus platform provides us the ability to efficiently express these antigens and also our ability to engineer this expression system to assemble the protein antigens into a suitable format for both stability of the antigen as well as ensuring that the size of the particle is appropriate for immunogenicity. And that really helps us in superior immunogenicity and better functional immunity because the epitopes are concentrated and are able to generate relevant antibodies. The manufacturing platform again allows us to produce products which are properly folded, modified, and then unlike traditional vaccine manufacturing or conventional vaccine manufacturing, you really don't need to grow an actual pathogen. Everything that is used in the development and manufacturing of vaccines utilizing this platform is recombinant. It's completely grass. We use SF9 cells and baculovirus expression system and genetic sequence of the protein antigen of interest. So unlike a conventional vaccine manufacturing facility where you will need to have a separate manufacturing facility for, say, rabies, a separate one for Ebola, a separate one for Zika in case you are making these in case you are manufacturing these products. In our case, we use the same manufacturing facility for all of the products that I've talked about so far. We make rabies, influenza, MERS, Zika in the same manufacturing facility. Also, that we really don't need a higher BSL-3 grade facilities. Unlike the conventional vaccine, if you are actually making vaccines utilizing the pathogens, you need to have very strict controls and higher BSL level facilities to be able to manufacture those products. We make our products again in a regular grass level manufacturing facility, obviously with appropriate classification of areas for GMP requirements. And again, I think I've already touched upon that same manufacturing is utilized to manufacture all of our products. For upstream, it's all single-use process. Up to production bioreactor, we have also implemented a single-use centrifugation system. And then for downstream, it's what we do is we dedicated columns for different products. Because as you know, same column can be used over, in some cases, hundreds of cycles for purification. We don't turn around columns for manufacturing different products. We have dedicated columns for every product, and then when we run a campaign for a particular product, the dedicated columns are utilized. The rest of the hardware is the same for all of our products in downstream as well. With that, I think I come to the close of my presentation. Thank you very much for listening, and I'll be happy to answer any questions just towards the end of the presentation. So, Amélie, over to you.
Thank you so much, Atin. Once again, I would like to remind our viewers that there will be a live question and answer session at the end of the presentation. Please feel free to send in your questions at any time during the presentation via the Questions tab located at the right-hand side of your screen, and we will go through these at the end of the webinar. Now, please allow me to introduce our second speaker, Amélie Boulais, Vaccine Platform Marketing Manager at Sartorius Stedim Biotech. Amélie Boulais started her collaboration with Sartorius in 2008 and as application specialist for the Purification Technologies division responsible for Southern Europe. In 2012, she became membrane chromatography and virus clearance project manager covering Europe. She was focusing on polishing and virus clearance. She became part of the process development consultant team in 2015, supporting and advising actors of the biopharma industry in their development from upstream to final filing operations. She joined the bioprocess platform team in 2016, taking over the responsibility of vaccine applications. She is analyzing the trends of the vaccine industry in order to develop fully scalable platforms with the required associated process development support and related services for vaccine applications. Amélie graduated from ENSTBB Biotechnology Engineering Institute of Bordeaux, France, and holds a biotechnology engineer degree. Welcome, Amélie.
Thank you very much for the introduction. So thanks also, Atin, for this great presentation around this nanoparticle technology platform. In this part of the webinar, I would like to discuss how we can develop and implement manufacturing platform. So I will be more focusing in this part to this part of the platform. So when I started as vaccine platform marketing manager at Sartorius, I was given the difficult task to assemble a single-use platform for vaccine customers. And if you ask me the question, do you believe that we can have a single-use platform for vaccine application? My answer would be no, we can't. We need to have multiple platforms. And this is logical because when you look at vaccine manufacturing, we know that each process will have its own reality. We have product-related consideration, especially if we are working with the pathogen. We have specific safety requirements, and we also have the constraints from the cell that is used. The advantage of using technology platform like the nanoparticle one or the virus-like particle one is that you're not using this pathogen anymore. So we can really have this platform approach for the manufacturing. So for me, the definition of a manufacturing platform, it is a toolbox of pre-selected and integrated technologies and services to support the development and the production of your vaccine along the drug life cycle. The toolbox part is important because even if we are working with a technology platform like nanoparticles, for example, we need some flexibility, especially for the downstream part. So the first dimension of the platform will be the process itself, from upstream to downstream and form and filling. But the second dimension also that I want to discuss during this presentation is that we need to put in place this platform over time. This is not a platform that you can implement directly into manufacturing. We need to implement during early stage process development, meaning that you need to have the right tools that are scalable very early on in your process development to make sure that you can go to this manufacturing platform. The benefit of such a platform will be that you can gain time to market. You do not need to reinvent the wheel every time that you develop your process for a new indication. You can reduce cost, you can reduce risk, and you can gain flexibility. You can use the same manufacturing platform as described by Atin for different indications within the same manufacturing facility. So let's enter into more details. This is the platform that we have developed on the upstream part for viral vector, virus-like particles, and nanoparticles. As you can see, this platform will be coordinated around a single-use bioreactor. So that's our biostat SCR. It's a stir tank single-use bioreactor, which is the heart of this platform. But of course, you need a lot of things around that. We can integrate process analytical technologies such as a capacitance probe. We can integrate the full seed train. We can integrate process development tools that I will describe in a few minutes. And of course, all the media preparation parts, including ready-to-use media, but also option to prepare your media on-site, including single-use mixer, filters, and so on. Just after the upstream part, we can of course have different options for the clarification. And what Atin already described is that of course we can use different technology depending if your product is extracellular and you will find it in the supernatant, or if it's intracellular and if you have a cell lysis, that's going to be a different approach. And last but not least, we also try to think about analytics, which are key for this kind of application. And we also have a virus counter that can be used for this type of applications. So let's enter into more details. So when we talk about a manufacturing platform upstream, we need to start in process development. And we cannot talk about process development without discussing design of experiments. Here on this slide, I have added an extract from the ICH-Q11. Small-scale models can be developed and used to support process development studies. The development of the model should account for scale effect and be representative of the proposed commercial process. This is exactly what we have done with our Ember portfolio. So the Ember product line, this is what you have on the picture here, is well known from the industry now and it's well adopted. For those who are not familiar with it, we are talking here about multi-parallel platforms of microbioreactors. They can be either 15 milliliter or 250 milliliter. You have here a picture of the 250 milliliter. So we talk about platforms of connected bioreactors that have the same control software. So we have integrated multiple bioreactors. If we look at the picture on the left, this is the high throughput system where we have 12 or 24 microbioreactors. We have a follow-up of pH, temperature, and OD on each of those bioreactors, so on the individual ones. And we have a fully automated sampling of in the bioreactors. So it really simplifies design of experiment. And we have a smaller version which is called the Ember 250 modular, which is up to 8 bioreactors in parallel. The difference here is that the pipetting and the sampling will be manual. The advantage is that these controlled microbioreactors, they are directly scalable into the largest 2,000 liter single-use bioreactor. And they are even scalable to any type of tier 10 bioreactor, including the glass vessel. So it means that if we look at our universal range of bioreactors that are from 0.6 to 10 liter working volume, this also scales. So how does it translate into our portfolio and into the platform? So this is what we have available today. We have the 250 milliliters for process development. We are going up to the 2,000 liters. So it means that we have a fully scalable platform and we also have the seed train there. We have the same geometrical design for all the vessels and we have a consistent mixing and casing strategy. It will simplify your scale-up but also your scale-down studies. You can easily switch between conventional and single-use bioreactor, and it of course mitigates risk during your process transfer. We have verified the performances of this platform with different applications. So we have demonstrated this capacity of the Ember system to be scalable to the large scale with CHO cell application, for example, for monoclonal antibody production. So what you can see here on this chart is the scalability of the platform at different scales. So the 250 milliliter bioreactor, a 5 liter glass vessel, and the 2 different scales of our single-use bioreactor, so the 50 liter and the 1,000 liter STR. On the chart, you can see the viable cell concentration, the cell viability, and the product concentration. You can see that we have an excellent scalability. Unfortunately, I do not have those data yet to illustrate insect cells, but we know it works. We have customers using the Endo system for those applications. And we also have customers such as GSK or Zoetis working in the vaccine field who have been showing scalability from the glass vessel, small-scale glass vessel, up to the biostat SCR for viral applications, so virus production, either in suspension or in adherent cells. And those that have been also published during conferences. So if we want to look at SF9 cells into more detail to also illustrate what was explained before by Atin, we know that it's possible to grow insect cells into single-use bioreactors. If you have interest into some details, you can take the link that we have here on this slide, or I can also send it to you later on if you have interest. And you can download an application note showing that the biostat, our rocking motion bioreactor, can be used for cultivation of SF9 cells in serum-free chemically defined. And one of the challenges of insect cells is that they have a higher oxygen demand compared to mammalian cells. And we see that our single-use systems are able to support their growth. So I gave you here a short overview of what we can do around single-use technology to produce nanoparticles, proteins, and viruses. But however, we still have a limitation, because when we are using those multi-bioreactor platforms like the Endo platform for process development, or if we use our single-use bioreactor including process analytical technologies, meaning inline sensor, we're going to generate a lot of data. And do you have the capacity today to integrate and analyze all those data? How can you drive actions from these data? Well, this is why we also have decided to integrate software into our platform. So I will just do a highlight on 3 of them. The first one is the software for design of experiments. So it all starts with planning the maximal informative experiment and understanding your design space. So the software model will allow you to get it right from the start with a very comprehensive and easy to use design of experiment and quality by design solution. Then when we go more on the manufacturing side, you can use the Simca software that can help you to dig out valuable and knowledgeable building information in your historical data. So this is where you can truly get into the depth of the data, process and product understanding, and it will enable you to see what other people don't. And the last one is the Simca online software. So it takes the power of the Simca, but the idea is that you put it directly on your production line, and it will let you know when something is about to go wrong. So before you get a process deviation, the Simca software will tell you, and you can avoid deviation before it happens, thus saving your batch. And this is a clear competitive advantage because by using most advanced analytics in your daily operation, you can really have prescriptive analytics. So the key point here to remember is that data analysis truly creates value when it drives decision making. So it's not only about the equipment you have, but also on the solution that you have to really analyze all those data. So with this, let's move to the downstream side. So we also have a platform for the downstream part. This is something you can apply for viral vector, virus-like particles, and nanoparticles. Here, I would say that the image I'm showing you is more like an example of generic process. A process can include filtration steps. It can include tangential flow filtration with ultrafiltration and diafiltration. And we also include here membrane chromatography. I will enter more into the details of all those technologies. But the main point here to remember is that the downstream process will really depend on your product. So as Atin explained, we can have nearly a true platform for upstream, but it's going to be different for downstream because there is not one-fit-all solution for complex products such as viruses or virus-like particles. We see that in all processes we will see the same type of technologies. And on those new generation of processes, we use filtration, we use chromatography, and we use tangential for filtration. However, the number of steps, the order of the steps will be different. And usually the shorter is the process, the better is the recovery. So we try to have highly efficient process steps. So let me give you an overview of the different options that we have within the downstream process. Let's start with the clarification. Usually when looking at clarification filters, we have two big options. The first one will be to use what we call depth filter. Depth filter, like the Sartorius filter we have in our portfolio, are cellulosic-based filters, and you have some inorganic adjuvants such as diatomaceous earth. The big advantage of such filter is that they have the ability to retain a big biomass. However, this diatomaceous earth also has the ability to retain small contaminants like DNA. It is usually an advantage, but when you're working with complex particles such as viruses, virus-like particles or nanoparticles, it could be that they have a strong negative charge and that they could absorb onto the filter. So the recovery of your product really has to be verified during small-scale trials. And the alternative to it is to use what we call a polypropylene pre-filter, like our Sartorius PP3. The advantage of such a filter is that there is a very low absorption. It's going to maximize your recovery. However, it has a lower capacity than a classical bed filter. So you really have the balance between those two options. And they can both be used for extracellular products, so just to clarify the supernatant, or for intracellular products if you have a cell lysis. But if you really go to intensified processes or high biomass, we have a third option, which is the Kstep system that we mentioned, that Atin mentioned earlier. Basically, the system can be used for high biomass removal. It's not working extremely efficiently when there is a cell lysis, so it's more for products that is directly into the supernatant. So we have here a fully single-use and closed system. It is cGMP compliant. This is of great interest if your particle is bigger than 0.2. If it's the case, it means that you cannot do any serial filtration and you need to work in a fully contained environment. And the Kstep can provide this, of course. So we have two systems available at two sizes. So we have a small-scale system. So that's a 400 milliliter chamber volume. You can handle 40 to 80 billion cells per cycle, meaning that basically you can harvest a 200-liter bioreactor in 6 to 8 hours. The largest system is having a 6-liter chamber volume, and you have 600 to 1,200 billion cell capacity per cycle, meaning that you can clarify a 2,000-liter bioreactor in 6 to 8 hours. It's quite complicated to explain the principle without showing a video, so if you have interest in the system, I can probably show you a video. Just contact me directly if you have questions around the principle, but basically you have this fully single-use chamber. It's a simple, robust, and scalable single-use centrifugation process with very low shear. So it means that you will keep your cells viable during the clarification. It's going to be more than 90%. So this system is also used for cell therapy processes, for example, where the cell is the product of interest. In our cases, we can also, of course, collect the supernatant containing the virus-like particle or the nanoparticles. You can use it also as a real continuous process, and it has the ability to handle large volumes with high cell density, up to 200 million cells per ml. So that's really a technology that we will advise if you work on intensified processes. Following this step, if your product is smaller than 0.2, we can of course implement a membrane filtration step to make sure that there is no cell or cell debris that go through the clarification. Here, as you know, at Sartorius, we have a very large range of filters. So we have two filters, I would say, that work very well for those type of applications. So we have the Sartorius XLG, which is a polyether sulfone filter with a combination of 0.8 and 0.2 micrometer. And we have the Sarto brand P, which is a cellulose-based, an acetate cellulose filter with a combination of 0.45 and 0.2 micrometer. Those filters usually work well for viral filtration, but of course we have teams of application specialists that can come in your facility and screen different filters to really find the best option for your product. We see that the best filter is really product dependent. And one of the key technologies that we have in our portfolio that is very interesting is what we call membrane adsorbers or membrane chromatography. So Atin here has described a process including chromatography resins, but they can have some limitation. When you are working with very big molecules, you can have steric exclusion effect. It means that your particle will be able to access the ligands that are around the bead, but they cannot diffuse inside the bead. So somehow you will not use 100% of the capacity of your resin because you lose what is inside your beads. In comparison, when you work with membrane adsorbers, those are cellulosic-based membranes with a pore size between 3 and 5 micrometers. Thanks to this big port, you have very high flow rate, but also all ligands that are grafted onto the membrane will be directly available. And if you compare the capacity of a membrane and of a resin for viral application, we usually have higher capacity for membrane. So the dynamic capacity can be up to 10 times higher with membrane than resins, or even maybe more. And those membranes are available with different ligands, so you can find a classical anion exchanger, such as quaternary ammonium, like a Q membrane. We have the S membrane, which is a cation exchanger. We have a hydrophobic membrane, which is a phenyl ligand. And we have a salt-tolerant interaction chromatography ligand, which is a salt-tolerant anion exchanger. And we just recently launched, and it's not mentioned here, a sulfated cellulose membrane, which is a pseudo-affinity ligand that can be used for influenza purification, but also for other types of viruses such as vaccinia virus. So it has some interest in the viral vector applications as well. And those membrane adsorbers are available at different scales, from 96 well plates and small devices of 1 milliliter around, up to very large volumes. So we talk about volumes of membrane to have the same wording than the resins. So it can be used for the capture of large molecules, but also for contaminant removal, so polishing in FOFO mode for DNA removal mostly. And those can be used in a single use or in a completely reusable way. It depends really of your application. The advantage of those devices is that they are delivered ready to use. You can autoclave them if you want to sterilize them. And that's it. And last technology, but not least, that's the tangential flow filtration. So you can use it to concentrate and diafiltrate your product. And I think what is important to consider is, like, when you use large setups, such as 100 kD or 300 kD, depending on the size of your particle, we can really consider it as a purification step because you can remove a lot of contaminants during the diafiltration. So we have a range of products that is available from 50 square centimeters up to 7 square meters, and that can be used for development and production. And as you can see on the picture, we have a range of classical stainless steel system available that can be used also with bags to have kind of hybrid options to simplify the cleaning of the tanks. And we also have fully single-use systems where we're going to deliver a gamma irradiated cassette, a gamma irradiated recirculation bag, all of these included in a loop a recirculation loop that will include all the sensors, meaning that you can have a fully single-use tangential flow filtration step that will be fully automated. And this is key when you are working on a manufacturing platform. Like if you have a multi-product platform, it's going to simplify the switch from one vaccine to the other because you don't need to validate the cleaning. And the last technology that I would like to introduce before my conclusion is the Amber approach that we have applied to tangential flow filtration. This is the first time we are trying to kind of Amberize our downstream application. We have developed a high throughput solution for process development of USEF, so tangential flow filtration. So we have developed a system where you can have up to 16 automated parallel trials of tangential flow filtration. You're going to have a 10 square centimeter cassette per channel, and you have a minimum recirculation volume of 5 milliliters. It means that you can use this system to really screen many different buffer conditions, for example, for your final formulation. The system has been launched with 10 kD and 3 kD cassettes, but the largest cutoff will come very soon, meaning that you can really fully use this very soon for vaccine application as well. So to conclude this part of the presentation, I would like to highlight the benefits of using single-use technology for this type of platform. The advantage is that you can get your facility running much faster, 12 months in average instead of 24 months for a stainless steel facility. You have a faster turnaround, meaning a higher utilization. You have more flexibility. You have a footprint reduction. You can reduce your cost also in your CapEx, and usually between 25 and 40%. But if you want to have more details on that, we also have a service of cost modeling where we can really identify what would be the cost reduction for your own process depending on the plan that you have for production, and we can drive you and guide you to a fully single-use process, a hybrid process, or even stainless steel process depending on what's going to be cost efficient for you. We're going to have, with single-use, fewer CO2 emissions and reduce cost contamination. And the benefit of a manufacturing platform is that you're going to decrease time to market, you will reduce costs, mitigate risk, and gain flexibility. And if we combine all of this, we see that single-use platforms are fully adapted to do this new generation of vaccine processes where we do not produce the pathogen anymore, but when we rather think about a technology platform. So thanks a lot for your attention, and I think now we are ready to take some questions.
Thank you so much, Amélie. Now we will begin the question and answer segment of the webinar. Once again, I would like to remind the audience that they can still send their questions via the questions tab on the right-hand side of the screen. Let's start with the first question. How quickly can you respond utilizing this platform in case of emergency, new pandemic? Atin.
Sure. So, yes, I think I had mentioned that it could be quick, but how quick? We've demonstrated for a couple of products that it could be done anywhere from 90 to 120 days. There were a couple of examples, and these are published in peer-reviewed journals where we were able to do, when I say we, with Novavax, we were able to do influenza vaccine, a pandemic influenza vaccine. We were able to get the GMP-grade material ready for human use from the publication of gene sequence in 90 days. So the day the sequence was announced and we started synthesizing the gene, from that day to having a GMP-grade material ready for human use was 90 days. Same exercise for Ebola was completed in 120 days. So that's the time frame we are talking about, 90 to 120 days, with demonstrated examples while in case of conventional vaccine, I think I mentioned earlier, it could take anywhere from 8, 10, 12 months, because once a pathogen is identified, it's isolated, then it's adapted so that it can actually be used as a vaccine strain. So it takes a long time to develop a vaccine candidate with conventional technology.
Thank you, Atin. Do you have data about the purification of nanoparticles or virus-like particles using membrane chromatography? What kind of performances can we expect there? Amélie.
Yes, so we have extensively evaluated Sartorius on viral application, so viral vectors, inactivated viruses or live attenuated viruses. So we have a lot of data available there that you can easily find. For example, around adenovirus purification, we've been showing that we can have up to 10 to the power 13 virus particle bound per milliliter of membrane. So we have a lot of data, and we have experience in purifying influenza, adenovirus, AAV, lentiviruses, rabies, or different types of viruses. And I think the biggest use that we have today in the industry and commercial process for vaccines is mostly for the polishing step for DNA removal. And we are building this data for the capturing part. Unfortunately, I don't have data to share about nanoparticles directly, but we do have a few publications available about the purification of virus-like particles. And basically the experience that we have is that large particles that have the size of the virus can be easily purified with Sartorius as long as the ligands that we have are fitting well with this type of application. And we can, of course, organize trial with our application specialist team that can come directly in your lab to generate those data.
Thank you, Amélie. What are the regulatory challenges in utilizing the same facility for multiple vaccines? Atin.
I guess they are, in our case, since we are not really dealing with pathogen or pathogenic material, these are the usual challenges that are there for any biologics manufacturing facility, multi-product biologics manufacturing facility for single use. We have to go through changeover. We need to have appropriate systems and processes in place to change over from one product to another, line clearance, and so on. However, having a single-use platform extremely simplifies the regulatory challenges, because the product contact surface is basically brand new. We change over in a single-use manufacturing facility. So for upstream, since everything is single use, it's quite simple. In case of downstream, as I mentioned, we use dedicated columns. The only thing which is sort of used across multiple products is really the hardware. So we just need to do the appropriate cleaning and line clearance activities and appropriate checks and balances in place to avoid any cross-contamination through the equipment hardware. So with this technology, again, completely recombinant, no pathogenic material, majority single use or dedicated, it's the usual, systems and processes that are required for any regular biological product like, even therapeutic proteins and so on. So much simpler compared to if you were doing a conventional vaccine.
Thank you, Atin. Is anybody using your single-use platform for commercial manufacturing or clinical lot production? Amélie.
Yes, I think that most of the products that have presented in the platform are used for production or commercial, for clinical like load production or commercial manufacturing of viral vectors or vaccines. The Biostat STR single-use bioreactors, it's a platform for a lot of our customers, and we can share some bibliography, including customer reference if interested. Our filtration technologies are widely used too. Sartorius, the membrane adsorbers are already validated on existing vaccine processes. The oldest application is DNA removal, so more polishing, but it's more and more used to capture the molecule of interest. However, we have a limited number of customer reference for the use of the entire platform. We have one case study that is quite new that I can share with you, where a company called ABL in Europe has implemented the entire platform for viral vector production. So it's a contract manufacturing organization, and they have implemented a suspension train for viral vector production with a focus on oncolytic viruses with our platform. So it includes the single-use bioreactors as well as the FlexAct system that is able to monitor the clarification and the tangential filtration steps. So they have implemented all our technology, excluding of the chromatography skid, but Sartorius could be used in the skid if needed. And this platform, I think, is in place since the beginning of the year, and it was implemented within a short time frame of 11 months for a scale of 200 liters, and they know they can produce 15 to 20 batches per year with such a platform.
Thank you, Amélie. Can different reservoirs of dengue virus vaccine developed using the recombinant nanoparticle technology?
Yes, it can definitely be developed. I think the challenge with dengue vaccine is really the level of response that you get across 4 serotypes. So yes, it 4 serotypes of dengue vaccine, then 1, 2, 3, 4 can be developed utilizing the nanoparticle technology. Could be basically the antigens can be expressed on the VLP surface, or potentially, again, I've not really studied dengue virus epitopes or antigen of interest for making a dengue virus. Most of the dengue vaccines right now being developed are live attenuated, so they are using whole pathogens. But assuming that there are certain specific antigens, either one or multiple, which have been or can be identified for protection, for protective titers, then those antigens, if there are multiple antigens, they can be expressed on the surface of a VLP. If there is a single antigen, that it could be expressed and assembled into a protein micelle format. Possible, but just thinking about the way dengue vaccine works, especially with the experience of Dengvaxia from Sanofi, I think the critical aspect for a dengue vaccine would be to ensure appropriate balance of response across all 4 serotypes.
Thank you, Atin. Before I finish the webinar, I would like to ask our presenters, Atin and Amélie, if they have any closing remarks. Atin.
Not really. I would just like to thank everyone for listening, and we believe that the recombinant nanoparticle technology has huge potential, and the products that we have in the pipeline, we believe that we'll be able to make a significant impact on global public health, and hopefully also to be able to respond. I mean, you don't hope that something bad happens, but in case something comes up, that with our technology we will be able to respond to be able to control any potential pandemics.
Thank you, Atin. Amélie, do you have any closing remarks?
Yes, I would like to thank the audience for attending the webinar, and thanks a lot, Atin, for this great presentation. It really shows that those new generation of processes and technologies will really help to develop new indications and to respond fast to any potential pandemic. And as a technology provider, we are committing to provide the right technologies to the industry, and do not hesitate to contact us if you are developing a platform, if you have a lack of technology, so we can help you to develop the right manufacturing platform that can help you to turn your technology into a real manufacturable process. And yeah, just a message, it's just contact us as soon as you have a specific need, because we need your feedback to be able to develop the technologies that will be able to handle the processes of tomorrow. Thank you.
Thank you. I would like to take this opportunity to thank our presenters, Atin Tomar, President and CEO at CPL Biologicals Pvt. Limited, and Amélie Boulais, Vaccine Platform Marketing Manager at Sartorius Stedim Biotech, for sharing their knowledge with us. I would like to remind our audience that they can view this webinar on demand by visiting biopharma-asia.com. Thank you, everyone. Have a great day.